Hi there. This is DNA segregation part 2. In part 1, I discussed some of the basics about how bacteria divide their DNA among the daughter cells, as well as some variations on that, and then talked about mitosis and some other methods of separation of chromosomes. In this video, I'll talk more about the evolution of ways to segregate DNA. I'll also talk about some different types of changes to DNA levels and movements of certain pieces of DNA, but I'll discuss that in a later video as well. The evolution of how to separate chromosomes equally among cells began back here with bacteria, where bacteria have bacterial fission, so they do move their chromosomes around using proteins that are attached to the cell membrane. Some eukaryotes actually divide by fission also, so they're sort of a progression from bacterial fission into eukaryotic fission, and then a true mitosis and cytokinesis. And then eventually meiosis too, which is sort of a recombination division and reduction division, in addition to a mitotic-like division as well. And then, as I mentioned last time, that some eukaryotes have a mitosis that's astral, some are anastral, some the nuclear membrane remains intact, although many have the nuclear membrane disintegrate and then reassemble around the chromosomes during telophase. While many eukaryotes have a haploid stage, organisms can only undergo meiosis if they have a diploid phase, where then they duplicate the chromosomes, have a tetraploid stage where they have 4 sets of the chromosomes, and those are then distributed after recombination among the 4 gametes that result from that meiotic division. So again, there's a progression from bacterial type division of the chromosomes. So that's 1 copy of the genome per cell, usually. And then there's mitosis with intact membranes, and then meiosis can be added onto that, and then a donut-like nuclear membrane with meiosis, partial dissociation of the nuclear membrane with meiosis, and then mitosis with complete disintegration of the nuclear membrane, although, as I said, those pieces of the membrane still exist within the nucleus, except in the case of basidiomycetes, where a new membrane is built up, and then you have meiosis there. Some organisms appear to have lost all of their sexual stages, and so in some cases that means that there is no meiosis, and in other cases there are meiotic divisions, but then there are hermaphrodites that form both types of gametes in one organism. So a lot of variation among organisms, especially among eukaryotes. So if we superimpose these on a phylogenetic tree, so here are bacteria and archaea, there's the root back around 4.0 to 4.2 billion years ago. And then we go into the eukaryotes, consisting of the excavata, acrobians, the SAR groups, Stromatophiles, Alveolata, Rhizaria, Archaeplastida, then amoebozoans, and then the opisthocons, which include fungi and animals. So bacteria and archaea down here, as far as I know, exclusively divide by fission, and they separate their chromosomes in a similar way, attaching them to proteins. And in some cases, those proteins in the planktomycetes resemble microtubules, so there's a little bit happening already in bacteria here as far as moving towards mitotic type divisions. And then moving into the eukaryotes, which primarily have mitosis plus meiosis, although, as I said, there are exceptions. There are some organisms in the excavata that have binary fission or fission type divisions. There are things up here in the fungi which have also a fission type division, but they also have microtubules to separate the chromosomes. So there's sort of a in this basal group here some signs of this evolutionary process moving from a simple fission into mitotic type divisions and then eventually into meiotic divisions as well. So over here in this group, especially in the dinoflagellates, you have a couple of different things happening. You have anastral type mitosis evolving here with an intact nucleus that never breaks down. And the chromosomes are within the nucleus and just move as the nucleus is pulled apart by the microtubules. There are some dinoflagellates up here in the alveolata also that have astral mitoses, where you do have clear polar microtubules and kinetochore microtubules, and the kinetochore microtubules are attached to proteins that attach to the chromosomes, and then the nucleus itself is pulled into two pieces with those chromosomes. Down here in the excavata, again, there are fission type divisions down into some groups here, where the chromosomes are indistinct and the nucleus is essentially just pulled into two pieces. Now, up here in the fungi, the ascomycetes and basidiomycetes, you see the branch they came off of here is rather close to the alveolata, so some of this may be a very ancient character in the fungi, where in the ascomycetes, the microtubules form inside of the nucleus, and then the nucleus is pulled into sort of a torus shape and then pulled apart. And the chromosomes, which again are indistinct, you don't really see chromosomes like this, those are pulled to the poles, and then these nuclei are pulled to the poles of the main cell. In basidiomycetes, those chromosomes are actually ripped out of the original nucleus, and then new nuclear material assembles around those chromosomes. And again, the microtubules are pushing those chromosomes apart as the nuclear membrane is forming around them. So both of those conditions are rather unique among all organisms. So those are really stand out as being a bit different, and so on a different evolutionary path than most of the other organisms, most of the other eukaryotes. Because in most animals, you have this condition where you do have condensation of the chromosomes, so you can actually see them. And they have astromitosis similar to these dinoflagellates over here with astromitosis. But in this case, the nuclear membrane does break down, and then during telophase, it reassembles around the chromosomes. In amoebae, it's sort of intermediate between fungi and animals in that you do have microtubules initially forming inside the nucleus, so that's like fungi, but then eventually the nuclear membrane breaks down, or it breaks down enough that it looks like an endoplasmic reticulum instead, and then reforms around those chromosomes during telophase. In plants, the Archaeplastida, these have anastral mitosis, and so the nuclear membrane breaks down, the microtubule organizing center, essentially that's where the microtubules emanate from, and you have kinetochore microtubules and polar microtubules pushing those chromosomes apart during anaphase, and then in telophase the nuclear membrane starts forming again around the chromosomes. And then, of course, the cell wall is built up, or the cell plate in between, forming a cell wall between the two daughter cells. So there's quite a variety of mechanisms that have evolved to separate the chromosomes, going from here with bacterial fission and separation of the chromosomes using proteins to move those to the poles, and that character is pretty much constant through all of these, although the proteins differ from one group of organisms to another, especially as you get into the eukaryotes, where in some cases you have these microtubules that are only outside of the nucleus, and the nucleus remains intact throughout mitosis, through some where the microtubules form inside the nucleus, and then the chromosomes are pushed apart, and then new nuclear membranes form around those, or the nucleus never breaks down. And then you have a lot of cases where the nucleus structure does break down and then reforms around the chromosomes during telophase. And many of the eukaryotes have a meiotic division and a haploid stage. So I want to talk about that next. So in plants, the large plants that you think about, the trees and the flowers and things, most of their life cycle is spent as a diploid. That is, these are called sporophytes, or the diploid stage. The gametophytes, they also have a gametophyte generation, which is a haploid generation. And in lower plants, actually the gametophyte is the main form of those plants, and the sporophyte only lasts for a short time. And so there are organisms in between that spend about half of their time as sporophytes and half of their life as gametophytes. So I want to talk about this a little bit. So if you look at the plant here, here's the plant, which is in a 2N, 2C level. That is, it has a diploid amount of DNA and a diploid number of chromosomes. So that's where I was talking about this C levels and N levels. But in a flowering plant, you have two different gametophyte generations. One is the female side and one is the male side. So on the female side here, within the central part of the flower, there's a stigma, style, and ovary. And within the ovary, you have macrospore ovules. So that cell starts out tetraploid because it was diploid and then replicated its chromosomes in preparation for the meiosis, goes through the two divisions of meiosis, and then you have mitotic divisions of those cells. Only one of those becomes the egg cell, and then there are at least 7 other cells, sometimes more depending on the plant, which will eventually control parts of development and then form nutritive tissue as well. So that's the gametophyte, consisting of more than one cell in the gametophyte, in addition to the egg cell, which is haploid. And actually all the cells in that gametophyte are haploid. And on the male side over here, you have the anther and the filament making up the stamens. And here you have a microspore mother cell, which then undergoes meiosis. So it starts out, again, tetraploid. You have 2 divisions. Then you have haploid tissue here. And then that actually undergoes mitotic divisions, haploid mitosis, similar to what happens over here on the female side. And so you have 3 cells in this case. That's the gametophyte on the male side. And one of those is a tube nucleus that helps grow that pollen tube, so it goes down into the ovary of the female part of the plant, part of the flower. And then there are 2 sperm nuclei. One fertilizes the egg, and the other one fuses with a couple of cells within the female gametophyte and begins forming nutritive tissue for the embryo. In the case of animals, they don't have an extensive gametophyte generation. So in the female, the gametophyte is essentially the egg cell, just one cell. And in the case of males, the gametophyte is just individual sperm. So those are only one cell, also. So here in animals, the gametophyte generation, that haploid generation, is reduced as far as you can really reduce anything. So for plants over here, the gametophyte generation in many flowering plants consists of about 8 cells. In some, it's many more cells than that, dozens of cells in the gametophyte generation. And in the case of mosses and lower plants, the gametophytes are much more extensive than that. And the sporophytes are small, little organs that come off of the gametophyte. So it depends on what type of plant you're talking about. But in animals over here, the gametophyte and the sporophyte generations are quite different. The sporophytes are usually very large, and the gametophytes are very much reduced. If you go all the way back in plants to talk about very simple green algae, such as this one, Chlamydomonas, then most of the cells you find that are Chlamydomonas cells are haploid cells. So that's its haploid generation. And these haploid cells can divide over and over and over again. So you have haploid mitosis, which is very common in simple organisms, both plants and animals, and protists as well. So a lot of haploid mitosis going on here. They are of 2 mating types, a plus and a minus, based on proteins that they have on their flagella. Then at some point in time they'll fuse by joining their flagella to form a diploid cell, but only for a very short time. So that's the zygote. Then it will duplicate its chromosomes, becoming 4N, 4C, and undergo meiosis, forming 4 haploid cells again to repeat the process. In some fungi, you not only have a haploid generation, 1N, 1C, and a short diploid generation, but you also have a dikaryotic stage. So these are called the dikarya, where the 2 nuclei within the cell never fuse, or the only time they fuse is right before meiosis. So much of the life cycle of these organisms, which are the mushrooms, so down in the soil or in a petri plate, you'll see this kind of fuzzy material, which are all these mycelia or hyphae. And these are haploid cells. Then you get different mating types coming together to form the hyphal union. And this, then the cells in like a mushroom, most of the cells that you're looking at are N plus N. So they have 2 nuclei, one from each of the mating types that came together. They remain separate and they undergo mitosis separately, which is quite a trick. And I'll show you that in a few minutes. And only in the gills of the fungi. So if you look at a mushroom, the cap, and you look on the underside, those gills underneath, that's the only place that you have diploid cells occurring. So in that case, in the case of basidiomycetes, you have a club or a basidium, where the nuclei eventually fuse, undergo meiosis immediately, and form 4 haploid spores. Ascomycetes also go through a similar stage, but then each one of these haploid spores then germinates and forms a new hyphae consisting of only haploid cells. So those undergo haploid mitosis, and over here you're getting N plus N mitosis, and up here after the diploid you're only getting meiosis occurring. In ascomycetes, such as this one, Sordaria, but also Saccharomyces is a ascomycete, and so it can undergo this type of reproduction also, although it looks quite different. Again, you have these haploid hyphae here, one in red, one in blue. Those can form haploid conidia or haploid spores also, not only undergo mitosis to form these hyphae, haploid mitosis, but also form haploid spores that can rest for quite a while or be dispersed and then germinate again to start a new hypha. At some point in time, an ascogonium occurs in these once there's an anastomosis, these two different mating types getting together, and that forms a cell that is multinucleate. And all of those nuclei are haploid. Once there's a fusing, then the other mating type nucleus enters that cell, and those nuclei start dividing. So there's roughly equal numbers of the two types of nuclei in that ascogonium. At some point in time, then you start getting this budding off. You get cell division, where each cell actually then has one of each of the different types of nuclei. And it's amazing that they can actually do this, but then you have these N plus N or dikaryotic cells coming out of this ascogonium. Then at some point in time, in preparation for meiosis, this loops over or curls over and forms what's called a Crozier hook. And those two nuclei in the end of that hook come together as a 2N2C cell. So finally, or 2N2C nucleus and 2N2C cell. So you have a diploid cell there finally. That undergoes a process of meiosis, so you double the amount of DNA to 4C, undergo meiosis, and you have then 4 of those cells, each of which are haploid. Those then undergo mitosis, so you have haploid mitosis occurring, forming 8 ascospores in that ascus, and then those break open and then germinate again. Depending on the species, this can either be all of these are blue up here and all of these are red down here, 2 different mating types, or you can have them alternating. It depends on the type of species and how the mitosis, how the mitotic spindles form, the orientation of those within that ascus. So again, these are called the dikarya, both the ascomycetes and the basidiomycetes that I just showed, that have this dikaryotic stage where these cells down here and through here, those are N plus N rather than being haploid or diploid, they're dikaryotic. You also have cells up here that are multinucleate, and in fungi, there are many cases where you have multinucleate cells as well. So in these dikaryotic species, then mitosis is slightly different or very different than mitosis in a diploid or a haploid cell. So in the case of filamentous ascomycetes, you have 2 sets of spindles forming, again, within the nuclei. You then get stretching of those nuclei, carrying each set of the chromosomes to opposite ends of the cell. Then a new separation, a septa, not quite a cell wall because it doesn't completely close off the cell. Those are formed between the cells to form new dikaryotic cells. In basidiomycetes, they have a very unique way to divide the 2 haploid nuclei equally among the new cells. So, as I said, one nucleus breaks down, or actually the chromosomes are pulled out of those nuclei. In the case of one set of the chromosomes, one nucleus, the chromosomes separate just as they would in a similar way to ascomycetes. But the other one then gets pushed out the side through what's called a clamp. So they're actually microtubules pushing that out, and then the nuclei start reforming. In the case of this one nucleus pair, they're just separated as they were. Similarly, in ascomycetes, the other one migrates through that clamp, joins in, fuses with the other cell, and then gets deposited in that cell. and then you can see these clamps remaining after cell division. And as far as I'm aware, this type of nuclear division and movement through a clamp like this is unique to the basidiomycetes. That is, I don't know of any other eukaryote that separates its nuclei or chromosomes in this way. And I believe also that the dikaryotic type of lifestyle in ascomycetes and basidiomycetes, that's also unique among eukaryotes. And here are a few pictures of those clamp connections where the nucleus has actually moved through that clamp from one cell to the other. Here's a close-up, there's another close-up, and then you can see among these hyphae all these little bumps on the side of the cell, and those are the clamp connections where one of the nuclei has moved through that channel. If these were ascomycetes, then those cell walls would be absolutely smooth. You wouldn't see those little bumps. Okay, so we talked about mitotic type divisions in diploids and haploids and dikaryotic species. Now I want to talk about meiosis, which has two divisions and eventually winding up from a 4C level down to a 1C level at the end here. And the second division is much like a mitotic division, except you start from a 2C level and then reduce that to a 1C level. In the case of division 1, that's a very special type division in which the first parts of that are devoted to recombination. So there are a lot of crossovers are occurring during that first part of meiosis, and that's one of its major features. So organisms first evolved to be diploid, and that was so that they could carry two alleles for the same gene, so that if at least one of those worked, they could deal with a little bit of mutation in the other one. In the case of organisms that then developed meiosis, that was to also recombine combinations of alleles to hopefully come out with some, and always come out with some, that would work together more efficiently than other combinations. So that also allowed actually for larger and larger genomes and more complex organisms. So this part up here is primarily for recombination, and then this division differs from the second division in that chromosome homologs are being separated rather than sister chromatids. Sister chromatids then are being separated in division 2. So in prophase of division 1, first there is some condensation, but not condensation completely like in mitosis. And then during leptotene, zygote, and pachytene, you bring the chromosomes together and have the crossovers occur, and finally in the last stages resolve those crossovers and separate the homologous chromosomes. So here's meiosis in a nutshell. Here's the nucleus, which is at a 4C level up here. Leptotene, you're having condensation of the chromosomes. They're not completely condensed, so they look sort of zigzaggy or rough rather than the smooth chromosomes that you are used to seeing in mitosis. In pachytene, they're brought together, you get the crossovers, and by diakinesis here, all the crossovers are resolved. And then metaphase 1 separates the homologous chromosomes, and then into telophase to reform the nuclei. And then the second division is here. Now we're at a 2C level, but a 1N level of chromosomes. And you get, again, condensation of the chromosomes and separation of the sister chromatids, in this case, reformation of the nuclei and separation of the cells, and then you have 1C, 1N cells in that case in the end, which are haploid cells. So here are a few pictures of chromosomes during meiosis. So this is in prophase, and you can see the chromosomes look kind of zigzaggy or fuzzy, and then during crossovers you have this synaptonemal complex that brings those chromosomes together, so then you can get a crossover between the all four of the chromatids for that chromosome set. And if you contrast that with mitotic chromosomes, see how these look much more smooth. These are much more condensed than the chromosomes during meiosis. So if you compare mitosis with meiosis, initially you have diploid cells here. They replicate their chromosomes, and then the sister chromatids are separated. In meiosis, in the first division, the homologous chromosomes or homologs are separated, and it's only in the second division that the sister chromatids are separated into separate cells. So you have haploid cells with a 1C level and a 1N number of chromosomes. So if you look at the two together, then here's mitosis, where you have diploid cells duplicating their chromosomes and then separating the sister chromatids to come out with separate diploid cells at 2C and 2N numbers. Meiosis, the first division, separates the homologous chromosomes. So there is a duplication here, but then homologous chromosomes are separated. And this second division looks more like a mitotic division, except it's with haploid cells. They're at a 1N 2C level. Then you separate those at anaphase, then cytokinesis, and then you wind up with 4 cells at the 1C 1N level, truly haploid cells. So just to show you where the chromosomes are going and what they're called in meiosis, and I did this for mitosis before, so you can compare this diagram with the one on mitosis that I showed last time. So here you have the homologous chromosomes in a diploid. They get duplicated, so now you're at a 4C2N level. If you count the chromosomes, there's a 2N level, but there's a doubling, so you have 4 times the amount of DNA as in a haploid. Then during the first part of division 1, you have the chromosomes coming together, and you get crossovers, and then the first division is separating the homologs. So now you have each cell is at a 2C1N level. That is, it has a haploid number of chromosomes, but it still has a diploid amount or 2C level of DNA. The second division is similar to a mitotic division in the haploid cells, and you get a separation of the chromatids. They become chromosomes, and then you wind up with cells that are at the 1C1N level, truly haploid cells again. In meiosis, during that first division, getting the chromosomes together, some unique structures form. One of these is the synaptonemal complex, which consists of some lateral elements and then central elements. And the lateral elements connect to the chromatids, so 4 chromatids are brought together, and then the central element zips together. Looks just like a zipper, actually, in the electron microscope also. And then every once in a while on that synaptonemal complex, there's a protein complex called a recombination nodule. And this is where the recombination or the crossovers occur on those recombination nodules. So there can be from 0 up to many of these recombination nodules on the synaptonemal complex, joining 2 chromatids together in a crossover event. So here's an electron micrograph showing how those chromosomes come together. And you can see down here, the synaptonemal complex has zipped together these two chromatids, and then there's a recombination nodule sitting on top of that that's then joining the two chromatids together in a crossover event. So in the next few slides, I want to show just mitosis, meiosis, and then some other alterations of DNA amounts and chromosome numbers in eukaryotic cells. So here are the first two. Mitosis, where you start at a 2C2N level. Then during the S phase, build that up to a 4C2N level. Separate the sister chromatids during anaphase. Build up in telophase a new nucleus, and then divide the cells so you have two truly diploid cells at a 2C level of DNA and a 2N level of the number of chromosomes. And you can have haploid mitosis too, starting with 1C1N and winding up with 1C1N. So what's the advantage of being diploid? Well, it means you have two allele copies for the same gene. And that means one allele can actually mutate, and it will be somewhat protected by the other allele in that you'll have at least one copy. So this allowed genomes to get larger because the organisms could then survive better even while having some mutations, some mutant genes, which is an advantage over organisms that had only one copy of their genome, such as bacteria and haploid eukaryotes. So then in meiosis, you start from a 2C2N level, build that up to 4C2N in the S phase, go through the first division, which separates the homologs rather than the sister chromatids. And now those cells initially are at a 2C1N level, and then the sister chromatids are separated, becoming 1C1N cells, true haploids. And meiosis was an advantage for organisms, primarily because even if you had a large genome, you could experience some mutations that might actually be lethal, but if you can generate some crossovers among the chromosomes and put those lethal genes aside and just keep the best genes, then you could survive even seemingly lethal mutations through this recombination process. And again, that would allow genomes also to get larger because you could experience more mutations, even seemingly lethal mutations, and still have progeny that would survive. In eukaryotes, there's also a lot of polyploidy, and this is a process known as endomitosis. So in this case, you start with a diploid cell at 2C2N, replicate the chromosomes. Now you're at 4C2N, but then you don't undergo mitosis and wind up going through the S phase over and over and over again until you have a polyploid cell with a large number of chromosomes and a large amount of DNA. Now, plants, especially and animals, have also experienced polyploidy that led to speciation. So they maintained that polyploid form in those organisms and continued going. So essentially, you and I and most vertebrates we're at a tetraploid level of DNA compared to our vertebrate ancestors. I mean, way back, the chordate ancestors. And if you look at the ray-finned fish, they're actually octoploid with respect to their vertebrate ancestors. A lot of the storage tissue in plants become polyploid. So the cotyledons in beans, so the main part of the bean that you eat, there's a little embryo here, which is at a 2C level, but the main starchy parts of those seeds are between 60 and 120 C. So they're polyploid. And that's mainly to have a lot of starch available for the embryo. You also have storage proteins and a lot of DNA in which to feed to essentially the embryo during germination. The endosperm in corn or maize also is the same way. So most of that starchy part that you eat from the cornmeal, things like that, that's polyploid tissue, about 90 C in that case. One variation on this is called endoreduplication, where you get polytene chromosomes instead of a polyploid nucleus. So in that case, the DNA is replicated over and over and over again, but the DNAs do not come apart. So the sister chromatids, or whatever you want to call them, are all pressed against one another. So you get very fat-looking chromosomes, very thick chromosomes. And so those are at a multiple C level, but you still have a 2N number of chromosomes. And here are some these are from salivary glands of an insect. And these are polytene chromosomes, so this is just chromosome after chromosome copied one on top of the other on top of the other, and so you have these big bulky chromosomes. These puff regions are where the DNA is actually being transcribed, so that you have a large number of genes that are being transcribed in various regions of the chromosome. And that's one reason that polytene chromosomes exist, is to increase copy number, especially of some gene regions, so that you'll have extra copies from which to transcribe the RNA and make lots of those proteins. Polytene chromosomes also are found in plants, and it's primarily during embryogenesis. So you have this basal cell, here's the embryo up here, and then a region that's called the suspensor, sort of, like a placenta, where the embryo is then attached to the ovary of the plant, of the flower. And here in this plant, then the polytene chromosome are up to an 8,192 C level in some of the plants. So in polytene, then you have a high value for the C level. So there's a lot of DNA in those nuclei, but they're only at a 2N level because the chromatids have not come apart. So that process is called endoreduplication. There's another process that makes polytene chromosomes, but not all of the regions are replicated to the same degree. So in this case, you have a 2N number of chromosomes, and there's a duplication to a higher C level, but then this minus Y means some regions are not copied to the same level. And then there's the case of amplification, extrachromosomal amplification usually, although sometimes it can be on the chromosome. So in this case, you have most of the nucleus is actually at a 2C level, but some regions are copied extrachromosomally to produce extra copies, and I already talked about this with regard to ribosomal genes in an earlier video. They also have a 2N number of chromosomes, but then there are these mini pieces of DNA, mini chromosomes, because of the amplification as well. So in order to get polyploid nuclei, you go through G1, through the S phase to replicate the DNA, separate the chromosomes during G2, and then go through endomitosis, and then go around this many times to come out with a polyploid cell. You can get polytene chromosomes by a couple of methods. One is to go through the S phase but not separate the chromosomes, and that's endoreduplication. So you can go through this many times and create large polytene chromosomes, although some regions can be underreplicated. So in that case, you would go through the cycle this way, under-replicating certain regions and multiplying other regions. And that produces polytene chromosomes as well. For extrachromosomal amplification, then certain parts of the S phase, there are certain pieces of the chromosome that are replicated over and over and over again, extrachromosomally. And then there's other evidence for alteration of DNA amounts. So, as I already spoke about with regard to Vishafaba, the ribosomal gene copy number can vary within individual plants by as much as 12-fold. And copy number within a population can vary from 230 copies up to 22,000 copies and more. And if you look at histone gene copy number, we found also that it can vary by as much as 7-fold within an individual plant. So although a C value is supposed to be a constant value, that's what the C stands for, it's not necessarily that constant. So you can think of it more as an average value for that species. And just to show you in that Vishafaba, and I've shown this before when I was talking about ribosomal RNA gene copy number, within leaves, and this is I think 434 plants, the copy number goes from a low down here about 230 copies per haploid genome up to 22,000 copies of ribosomal DNA genes or ribosomal RNA genes up here. So that's a 95-fold variation within a population. I also at the same time measured the number of ribosomal DNA copies per haploid genome in cotyledons. And remember, cotyledons are 60 to 120 ploid. So you can actually multiply these up in the cotyledons, a lot more ribosomal DNA copies in there. And then with embryos, shoots, roots, etc. The average or mean of the ribosomal copies was always right around the middle here, which was in the thousands of copies, about 2,500 copies on average. And the way that those are changing is indicated somewhat by this southern hybridization here, where this 3.8 kilobase band is this 18S, 25S fragment here, and then all of this lateral bands is here in the intergenic spacer. There are these subrepetitive elements. And so these each vary by the number of subrepetitive elements. This average at 6.7 consists of 6 of those subrepetitive elements, and then going down to actually 1 all the way up to 23 of those. And so this is, and I'll show you in the next slides, this is indicative of the process that's leading to these differences in gene copy number. Additional evidence comes from doing genetic crosses of these bean plants, Vishafaba. In this one, in panel A, this is a parent and then the progeny from self-pollination of those. And you can see there are some differences in some of the progeny already. And then when I did genetic crosses, you would expect this parent and this parent to yield progeny that would be intermediate between them, and in most cases they were. And then I allowed these heterozygous F1s to undergo self-pollination, so then you got sorting out of those alleles. And in many cases, yeah, they looked like either the 1 or 8 parent, the pattern from parent 1 or 8. But in many cases, there were variations. So this one, you can see a variant of type 1, and the other two are a little more subtle, but there are differences in the intensities of the bands for the pattern 8 and also the heterozygous type. So this was an indication that although the genetics of this region were sort of Mendelian as far as how they were sorting out, they weren't completely Mendelian because you were getting some differences in the patterns, which indicated that something that was happening in somatic cells was influencing the patterns. So one other piece of information that helped was to do R looping, and this is where you mix the ribosomal DNA, which I purified, with the ribosomal RNA, and then denature the DNA, and then reanneal the DNA RNA mixture, and wherever the RNA fits, it will displace the DNA from that. So you can see these patterns in here of the 18S and 25S, and then going down through here another 18S, 25S, and another 18S, 25S. So you can follow these through. And what I was interested in is how large are the intergenic spacers, IGS1 here, and then a second one, IGS2 and IGS3. And are they arranged in groups? So all of the ones of the same size IGS, are they all in the same place? Or are they randomly arranged along the chromosome? And what I found is that they, as indicated here, that they were pretty much randomly arranged along the chromosome. And this pointed to unequal crossing over then. So both changing in the overall copy number and then the arrangement of the intergenic spacers. Both of those led to a conclusion that there was some sort of unequal crossing over going on in this region. And furthermore, since it happened within an individual, that is, that the copy number changed in a flower versus a leaf versus a root versus a stem in the same plant, it indicated that these changes were happening in somatic cells, that is, in non-gamete cells. So these were happening in cell divisions throughout the organism. And it turns out it's happening in yeast, and it's happening in humans, and it's a very common type of rearrangement in these repetitive regions, such as the ribosomal RNA gene region and many other repetitive regions as well. So here's an illustration of how that's happening. So these are the intergenic spacers on the left-hand side here. There's the 18S gene and 25S gene on bordering this region. So if you have 6 of these repetitive regions, subrepetitive regions, and 6 on the adjoining chromosome, and they misalign by one or more of those subrepetitive elements, and you get a crossover, you can generate new versions of those. One here gained and becomes 7 repeats, and the other one loses it, goes down to 5. This can happen over and over and over again, generating that ladder of bands. Like these ones up here, this ladder indicates a variable number of those subrepetitive elements in the IGS that you see up here. And each one of those steps varies by 325 base pairs. So this one has 6, 5, 4, 3, 2, and there's 1 down there, and then going up 7, 8, 9, all the way up to 23 in some cases. So that generates that variability in the intergenic spacer region. But over a longer range, if you look at the chromosomes here, where you have 2 chromosomes lined up, with regard to those ribosomal RNA genes, if they line up perfectly, then you get no changes. But if they align unequally with one another, that is, by one or more of those repeats in this case, so I've just colored them here for convenience, but if you line them up, so here's one chromosome, here's the other chromosome, if you get a crossover, the result is that one chromosome gains a large number of ribosomal DNA copies, and the other one loses a large number of ribosomal RNA gene copies. And this is as I said, this is a very common occurrence in plant ribosomal RNA gene regions, in humans, in yeast, and probably in most eukaryotic ribosomal gene loci. And I haven't shown it here, but we also did similar work on histone genes also, which are in multiple copies. And they do the same thing, but to a smaller extent, because they have fewer gene copies, but the same thing is going on. Unequal crossing over and creating differences in histone gene copy number in that case. And to reiterate, this is happening in somatic cells during mitotic division. So this isn't during meiosis to create gametes. This is happening in normal somatic tissue. Another interesting thing that has been found that in a different plant, Linum usitatissimum, which is flax, and flax is made into linen, so that's what linen is made out of, flax fibers. Flax seed you can buy to eat, and you can buy linseed oil for painting and other things. So it's a very useful plant crop. But 3 genotrophs have been identified, and these have the same genotype. They can be crossed, but the plants look somewhat different in a population. So there's a small genotroph, which is smaller, which has about 136 million base pairs of DNA in its nucleus, and then a plastic type, which is the normal type, which has about 150 million base pairs in its genome. And then there's a large genotroph, which is larger, and it has a little more DNA in its genome, in its haploid genome. And by a little, I mean 8 million base pairs of DNA extra. That's 2 E. coli chromosomes worth, so it's actually quite a bit of DNA. And this one loses about 14 million, and so that's about enough DNA for a yeast cell. So it's actually a lot of DNA. So here are the relative weights of the plant. One, the plastic type is twice as large as the small type, and the large type is larger than that. And again, even though the genomes are different, the plants have exactly the same gene makeup, and they can be crossed, and when they're crossed, they still make the small, plastic, and large types. And here are a couple of those plants. This one is the large type, and this is the small type, and then the plastic type would be about twice as large as that small one. So here's where it gets interesting. If you start with plants of the P type, the plastic type, and you use fertilizer containing ammonium, which gives a very rapid nitrogen nutrient to the plant, the plants will change from a P type to an L type. And when you get the seeds from that plant, all of the progeny are of the large type. So they're all larger plants, and they have more DNA than the parent plant did. If you grow them in phosphate fertilizer at a low pH, so it's not available, essentially, the phosphate is difficult for the plants to take up, then the P plant changes to an S type, and all the progeny are of the S type, and those S type breed true S types for several generations. If you give them phosphate fertilizer at a higher pH, then the P type remains P, and all the progeny are of the plastic type too. After several generations, both the L type and the S type, if they're not under any kind of stress, will revert to the P type. So this is just that diagrammatically. If the plants are not under stress and you start with the plastic genotroph, they remain as the plastic genotroph. If you grow them under stress, then they revert to a small genotroph, and all the progeny are of the small genotroph. And if you then put those small plants under no stress, then they eventually revert back to the P type. But if you give them high amounts of fertilizer, then they'll revert to the large genotroph, and they will remain in the large genotroph as long as you keep that selection on. But if you then grow them under non-stressful conditions and low fertilizer amounts, they will revert back to the P type and stay that way. All the progeny, that is, after many generations, will be of the P type. This was studied for many, many years. Also, if you grow them in large pots or small pots, these conditions can change also. But it was discovered that a lot of these large changes, which you can actually watch happening in the plant by going up the stem as you're changing the conditions, you can actually measure these differences, were due to transposons, which were moving pieces of DNA around in the plant genome. So to revert to the small genotroph, the transposons were actually getting rid of different parts of the genome. And when you reverted to the large genotroph, then additional transposons and sequences were being added to the genome. Another characteristic of plants is that as the tissues age, they actually lose DNA. So when an embryo is formed, it has a full complement of plastid DNA and nuclear DNA and mitochondrial DNA for that matter too. A young leaf has a full complement of DNA as well. But then a fully expanded leaf has already a large amount of degraded DNA. And so it's still forming RNA, transcribing genes and making them into RNAs, but some of the DNA has already degraded and is non-functional. And then by the time you get to an old senescing leaf, it has very little intact DNA. And that includes the mitochondria, the chloroplast, and the genomic DNA. So if you look at a leaf in the middle of summer that's fully expanded, its DNA is already degrading. And by the end of the season, when the leaves start changing color and dying, they have almost no DNA, no intact DNA. They have fragmented DNA remaining. And they can't form the RNA from those anymore. So, little by little, the proteins degrade and eventually the leaf and the different tissues die. So a set of experiments were performed, and it started long ago when I was actually a PhD student in this lab, where we were trying to take leaves from maize and from wheat and transform them, that is, add some DNA to their genomes. But we found that the leaves, even from very young leaves, the cells from very young leaves, were essentially non-transformable, and we didn't know exactly what was happening. But if you took embryonic tissue, that was transformable. You could inject some DNA in there and get it incorporated into the genome. So later in that lab, after I'd already left, they did a set of experiments to determine what was going on. So down here in a proplastid, in an embryonic cells, or very young, immature cells, the DNA was in full genomic state. That is, it was not fragmented. There were replicated forms of the DNA. All copies of the DNA were functional, low damage, low repair also. And the DNA function was for heredity and coding. mRNA was, and all the RNAs were being transcribed off of that DNA. So it was in very good shape in these embryonic or very immature cells. But if you looked further out on these leaves, in fact, if you looked at a younger leaf versus an older leaf, you could actually see the DNA become more fragmented. It had fewer coding copies. It was highly damaged, still low repair. And the DNA function appeared to be primarily nutritive, that is, recycling of phosphates and carbon, etc. So by the time a leaf is even, a few inches long, the DNA is already being degraded, and it's pretty much already transcribed everything that's going to be transcribed off of there, and then those mRNAs go on to make the proteins, are translated into proteins, and that's made in the leaf, and that's about it. By the time the leaf is fully expanded, the DNA is in such bad shape that there's not much transcription going on. Translation continues on the mRNAs that are there, but they have a half-life too, so eventually they start degrading, and then finally the protein degrades and the leaf is dead then. And this was true of all the plants that were examined, although to a different degree. So in Zea mays, corn, the degradation occurred very early in leaf expansion, and by the time the leaf was mature, there was very little DNA left. Arabidopsis was a little slower, but as the leaf was about half expanded, the DNA was highly degraded as well. And it was a little slower in Nicotiana tabacum, or tobacco, so that those leaves expanded and had most of their DNA until they were almost fully expanded, and then the DNA started to degrade after that. So the time scale down here is in terms of weeks. So only after about a week or so, the DNA in the maize leaves tends to start being degraded, whereas for Nicotiana tabacum, it's more like 4 weeks or so before the DNA starts to be fully degraded. This also happens with so-called evergreen trees, and if you notice, evergreen trees are losing their needles all the time. So the time scale down here would be in terms of months, or in some evergreens even years, but not more than a few years, and then those needles die. So they come out, they last a year or two sometimes, and then they die. And if you look into those leaves about midway through their lifespan, the DNA is beginning to degrade substantially. And this likely happens with the cells of all species, in that eventually there's a substantial amount of degradation that occurs in those cells, and they tend to then die and be replaced by other cells. In the case of vertebrate cells, like human cells, there are actually proteins that go around, like BRCA2 and BRCA1, that examine for DNA damage. And if there's DNA damage, then they signal p53 to kill the cell. So there are also mechanisms in animals that sort of check for a great deal of DNA damage. If there is DNA damage, then they signal the cell to be killed, or go through apoptosis or programmed cell death. So as you go back through these last two videos and slides, think about the implications of mitosis and meiosis, those divisions of the DNA, as well as other types of separation or segregation or amplification or replication of the DNA. So think of those in terms of humans and vascular plants, where the somatic cells are primarily diploid, the different processes there. In lower eukaryotes, where most of the life cycle is spent as haploids, so that's going to be slightly different in that they only have one copy of each one of those alleles for each gene locus. And then in fungi, some of which have two nuclei in the same cell, and some become polynucleate also. So that then causes other changes and characteristics of those cells. And I will discuss those in a little more detail as well in other videos. And then in some organisms that have parthenogenesis, that is, they just essentially create clones of themselves, and this happens in a lot of insect species and even reptiles and sharks and trees. And then there are organisms that have very low rates of recombination, and so with recombination you can certainly come out with different combinations of genes and try to eliminate some of the detrimental ones. If you don't have recombination, then that slows that process down and may increase mutational rates. So there are many ways to make copies of genomes, and you can make multiple copies, partial copies. You can have a lot of recombination, little recombination. You can be a diploid, a haploid, a polyploid, a dikaryote. a lot of different possibilities. And then sometimes environmental conditions and stress can lead to changes in mutation and or repair rates, and that can affect the amount of variation and diversity in a species. So that can lead to some problems in selection and survival in that species, depending on how much stress and changeability there is in the environment. So all of those things have led to different combinations of genes and mechanisms and species. And we'll talk more about this in coming videos as well. So this is the end of part 2 of DNA segregation. So I did talk mainly about DNA segregation, but then I've spent some time talking about alterations on the amount of DNA each cell gets, or how you get more DNA or less DNA in a particular cell. Okay, so that's it for now, and I will talk to you later. Thanks very much for watching and listening.